Hello, everyone. Welcome to my video tutorial for pseudo-bulk RNA sequencing analysis. We know that bulk RNA sequencing analysis is more accurate, more reliable when we perform differentially expressed gene analysis than single-cell RNA sequencing dataset. So methods were developed to perform pseudo-bulk RNA analysis for single-cell RNA sequencing data. So I'm going to make videos to show you how to perform pseudo-bulk RNA analysis. So today, my first video, I'm going to show you how to prepare the single-cell RNA sequencing dataset. Then we can use this dataset to perform pseudo-bulk RNA analysis in my next video tutorial. So the dataset I'm going to use for demonstration will be the GSE 132771 again. I frequently use this dataset in my video tutorials. This is a dataset that performed single-cell RNA sequencing analysis, not only in human but also in mouse for fibrotic lung. We can scroll down to see the dataset. You can see here. Here is the link that you can download the dataset. You can click the HTTP link to download the whole dataset. And also you can click the custom link to download the individual dataset. So for today's demonstration, we are going to download the human lung dataset. You can see here they labeled as AML1, AML2, and AML3. These three datasets are from healthy donors. And further down, you can see IPF1, IPF2, and IPF3. These three datasets are from fibrotic lung. If you click the download, you will download the dataset that I selected here. So I downloaded those datasets already, and it is in my HR folder. So we can go to my folder and have a look at those datasets. You can see in my HR folder, we have the dataset AML O1, O2, and O3, and also we have the IPF dataset O1, O2, and O3. Let's click the dataset and have a look at the files. Inside each dataset, you can see we have the barcodes file, features file, and the matrix file. If you watched my first short video tutorial, you will realize that when we download the dataset, the features TSV file was named as genes TSV. So you need to change genes to features, then you can load the data into R. So now we can go to R to load the dataset. First, we can load the libraries, thread, tidyverse, and also we set the set to make sure we can repeat the analysis next time when we run the same code. So now we can use the read 10 times function from the thread package to load the data. We will load the AML01 as the control one. Let's load the data. You can see we have a large DGC matrix data in the data window named as control1. Then we can use the control1 as a count to create a thread object. And we give a project name as control1 for this dataset. At the same time, we only keep genes that are expressed in at least 3 cells and the cells that have at least 200 genes. So let's create the thread object for the control1. So you can see now it changed to a thread object for control1. So we will do the same for control2. You see we have control2 thread object, then control3, So after that, we can load the IPF1, then IPF2. At last, we load the IPF3. So now you can see in my data windows, we have Surat object for each dataset, control 1, 2, 3, and IPF1, 2, and 3. So now we can create a list object to contain 6 datasets. Then we can use the control list to perform data integration. So let's create the list first. So now you can see we have a list object named the control IPF list in the data window. So I'm going to use the classic Surat integration method to perform data integration between control and the IPF dataset. So first we can normalize the data and find the variable features for each dataset. Let's run this code first. So we normalize the data for each dataset, and also we find the variable features for each dataset. To perform data integration, now we can select the integration features from the features that we find for each dataset. So let's select the features for data integration. Now we can use the features to find the integration anchors. Let's run. And this step takes time to run. It will take more than 10 minutes. Okay, we found the integration anchors. Now we can integrate the 6 datasets into one dataset, and we named it the control IPF combined. Let's integrate the dataset. So after integration, we can set the default array as integrated. Then we can perform cell clustering and visualization. So first we can scale the data, then run PCA. Next we can run UMap. After UMap, we can find the neighbors, then find the clusters. You can see I set the resolution as 0.05. But in this resolution, we have 13 cell clusters. So now we can have a look at the cell clusters. Let's zoom in. So you can see we have 13 cell clusters from 6 datasets. So we can also look at the individual dataset using the group by function. When we create the thread object, we set the project name as control 1, control 2, control 3, IPF1, IPF2, and IPF3. So those information should be found in the origin ident. We can use the group by as the origin ident to look at individual dataset. Let's run it again. And we can zoom in to see the cell clusters now. You can see we have cells from 6 datasets, and the same type of cells from each dataset are well integrated in the cell cluster. So next, we can annotate the cell clusters. So we can use the classic marker genes to identify the the cell cluster in human lung. So we normally use 4 markers EPCAM identify epithelial cells. CLDN5 is a mark gene for endothelial cells, and the collagen 1A2 is a mark gene for mesenchymal cells. Then PTPRC labels all the immune cells. So we can use the feature plot function to see the mark genes expression in different cell clusters. Let's run. And you can see a warning message here could not find the PTPRC in default search locations because at the moment we set the default assay as integrated, but found in RNA assay. If you set the default assay as RNA, then you don't have this warning message. So let's zoom in to see the cell clusters. You can see EPCAM is expressed in cluster 3 and 7. CLDN5 labels cluster 1 and 10. Then collagen 1A2 labels cluster 4 and 5. PTPRC labels the rest cell clusters. So the collagen 1A2 labels the mesenchymal cells. Mesenchymal cells contains fibroblast, parasite, and smooth muscle cells in the lung. So I'm going to use the fibroblast cell cluster to demonstrate how we perform pseudo-bulk RNA analysis and identify differentially expressed genes between control and IPF dataset. So let's use the fibroblast, parasites, and smooth muscle mark genes to identify fibroblast, parasite, and smooth muscle cells. Both NAM and DCN label fibroblast, RGS5 labels parasite, and DES labels smooth muscle cells. So we can run the feature plot again and zoom in to see the view map again. You can see both NAM and DCN label cluster 4. So cluster 4 should be fibroblast. RGS5 labels cells on the left-hand side in cluster 5, and DES labels cells on the right-hand side of cluster 5. So cluster 5 contains parasite and smooth muscle cells. Because we use very low resolution, if you increase resolution, you will separate parasite from smooth muscle cells. So now we know the cell types in each cell cluster. Then we can name the cell clusters as immune cells, endothelial cells, parasite, smooth muscle cells, and fibroblast. So we can use the rename ident function to rename the cell clusters. Let's run. And now we can use the dim plot to look at the cell clusters again. And zoom in to see the cell clusters. So now you can see we labeled the endothelial cells, fibroblast, parasite, and smooth muscle cells, epithelial cells, and the rest are immune cells. So we named the cell types for each cell. Now we can set the cell ident as cell types and create a column in the metadata to label the cell types for each cell. So we can create this cell type column in the metadata. Then we can use the view function to look at the metadata. You can see the row names are the cell barcodes. We have the control IPF dataset in the origin ident, count RNA, and unfeatured RNA. We have the cell clusters because we perform the integration and find the clusters at a resolution 0.05. So you can see the last column is the cell types we named for each cell. So next we can clear the UMAP in the plot windows. Then we can use dim plot and set the group by argument of the cell types to have a look at the cell canisters again. You can see the same UMap as the previous one. So let's save the integrated dataset as control IPF combined in my HR folder. Let's run. So we saved the integrated dataset. As I mentioned, I want to use the fibroblast cluster and demonstrate the classic HR function to perform pseudo-RNA analysis and identify differentially expressed genes between control and IPF fibroblast. So now we can subset the fibroblast cluster and save as an individual object. So let's use the idents as the fibroblast and subset the fibroblast cluster, and we create a new object as fibra, and we can use dim plot to check if they are the right cell cluster. Let's run. You can see now we only have fibroblast in this dataset. Now we can save the fibroblast object for next video tutorial. Let's save it. Okay, I'm going to stop from here for today's demonstration. So in this video tutorial, I prepare the dataset for next video tutorial. If you want to use the same dataset for practice, you can follow my video tutorial and I will post the code in the comments. Then you can prepare a dataset for next video tutorial. If you have your own dataset, you can also use your own dataset. So I hope to see you in my next video tutorial. Once again, please subscribe my channel if you haven't to do so. Thank you.